Let's begin with our first speaker, Dr. Katalin Karikó. She is one of this year's Canada Gairdner International Awardees for her pioneering work developing nucleoside-modified mRNA drug delivery, which is the foundational technology for the highly effective COVID-19 mRNA vaccines that we have all benefited from and have made this day possible. Dr. Karikó is Senior Vice President, RNA Protein Replacement Therapies at BioNTech, Professor at the University of Szeged, and Adjunct Professor at the University of Pennsylvania's Perelman School of Medicine. Please join me in welcoming Dr. Karikó to the stage.
Good morning. I am very happy to be here, very excited to talk about development of mRNA for therapy. I mostly talk about that 60 years which it took to develop RNA for an FDA-approved product after discovering RNA in 1961, 61 years ago. During this 60 years, a lot of things happened. There are some major things, but most of the time it's just the incremental improvement of mRNA, the delivery, and so on. So it started in 1961 when two papers in Nature described the label intermediate, which is carrying information from the DNA to the protein synthesis place, to the ribosome. And with this one, we know that the information flow in every cell in our body, as well as all free living organisms, is from DNA to RNA and to protein. So, of course, scientists, 1961, they couldn't synthesize RNA, so if that time, we wouldn't be able to make a vaccine, but scientists start to isolate RNA and start to analyze them. First, they looked at different structural elements. In 1972, they discovered that the RNA has a poly-A tail. In 1975, they discovered there is a cap structure. This was a very interesting product there. It's never seen before. There were modified nucleoside in it. And actually, this was the heydays when everybody started to investigating modified nucleosides. And so the SCAP is the Cap1 element, which if you get the vaccine from Pfizer-BioNTech, was incorporated during synthesis. But in this year, in 1975, several papers describing what is the importance of this cap, and one of the things was that this was required for translation. And by the end of that year, Bernard Moss and colleagues also discovered that there are viral enzymes which can put this cap on the mRNA, and if you get the Moderna vaccine, that was actually using these enzymes to cap the RNA. Other scientists further analyzed and further tested out what all of them was isolated RNA. So they could isolate RNA from reticulocytes, and these were globin RNA. And they translated it in lysate, cell lysate. They injected in frog oocyte. Those are large cells. It was easy, and they looked at for translation of these isolated RNA. And in 1978, two papers described that RNA, when it was wrapped in a liposome, then it can diffuse to primary cells and then generated and translated the liposome-encoded RNA. So this was the first time, really, when it was realized that maybe RNA could be used for some kind of therapy. And liposome was introduced by Branham in London, and a colleague in Dimitriadis in London, he delivered RNA to the cell. Prior to that, other scientists delivered with liposome plasmid DNA. And that was the paper we were reading in Hungary when I was undergraduate student and joined the lipid lab. And with my colleague, we isolated phospholipid, and we generated liposome, and we put in plasmid into it and delivered to the cell. So it is important for the students, start with something, because the rest of my life I was remember this experiment and was thinking about it. So it was not RNA, it was DNA, but delivering nucleic acid inside the cell by liposome was a very interesting thing to do. By chance, next step to make my graduate studies, my PhD thesis, I went to the RNA lab. It happened that my supervisor, who was an organic chemist, actually synthesized cap analogs and sent it to Aaron Shotkin and colleagues in New Jersey, because if you work with something, you need reference material. My role in the lab was to synthesize RNA. Those were short RNA trimers, and we made enzymatically, we made chemically, and this small RNA had antiviral effect. It was just discovered the prior year, 1977, Ian Kerr in London. And I worked together with János Ludwig. He was a graduate student. And again, telling you, students, who's sitting next to you one day will be, or rest of your life is your advisor, because every time I turn to János for advice on organic chemistry. So this RNA, this small RNA, actually naturally also produced in your body. But of course, our idea was to generate an antiviral compound. And because this molecule, when it is generated, can activate an RNase L latent enzyme, and that was believed that this is cutting up all of the single-stranded viral RNA, and this is how it expresses antiviral effect. It's not turned out everything like that, but we still had a problem delivering this small molecule and ending the funding for that. So that's how I get into the United States. And I still worked on this molecule, and we tried to develop it for antiviral effect, because in 1985-86, when I arrived here, the major problem was the HIV. But eventually, we had to use a different kind of RNA, not this small molecule, but long double-stranded RNA, which had some mismatch in it, and this was less toxic than the poly-IC used prior to that, and we treated HIV patients. So as you can hear here, that all in my life, I just work with RNA, whether I try to degrade with an enzyme or whether induce some interferon with double-stranded RNA, label the RNA, and so on. So I was very interested always in RNA. While I was working on these projects, scientists at Harvard University figured out how you can generate RNA from using a phage RNA polymerase. I don't have time to tell for everything. There is a lot of scientists worked on it. For example, making RNA in vitro in a tube. Scientists tried bacterial polymerases, but turned out that this phage RNA polymerase has very high fidelity, very effective, and you can generate RNA by adding a template, adding the 4 basic nucleotides, and the enzyme. In that case, they also added the capping enzyme, which Bernard Moss discovered, just I showed you. So they generated this RNA, which code for interferon, and scientists there injected to frog oocyte, cultured them, and they found that the medium had antiviral effect. So this was the first time it was demonstrated that messenger RNA made by design, injected into cells, can code for the generate functional protein. A lot of people get excited. And anyway, in the second part of the 80s, most of you were not born yet, but that was a very exciting time because the PCR was introduced. When the T7 and SP6 RNA polymerase was reported, the same year you already could purchase it. So scientists were excited because now that they can use the RNA for therapy. So here I am showing you all of the paper which was published in the 90s on mRNA. It was easy to follow the literature because every year there was just one paper. But when I try to follow what happens, many of these laboratories were not publishing more, not follow-up papers. For me, the most exciting was when it was shown that vasopressin mRNA could treat an animal, which was a Blatterboro rat, and which had some special disease. And injecting the RNA for 2 weeks, the animal was cured. So this gave me the idea that, you know, this mRNA can be used. By that time, I started in '89 doing some experiment, but this was very exciting for me. And you can see that there were publications on using mRNA for infectious disease against like influenza. And there were some papers where mRNA was used as a cancer vaccine. Only these scientists made follow-up and did a clinical trial, but most of the other groups stopped using. So what happened? What happened is the RNA had just problem. A very small amount of protein was produced, very short period of time, it degraded, and as we discovered also, it was immunogenic. So from the 90s, what happened is that continuous incremental improvement of the RNA, it purified a better cap, better process, and better formulation. That was happened. So everything was just incremental, and people usually they are not patient. And so, you know, many of the people left the field. So as I mentioned, I started in '89 working with RNA, and with my colleague, I started in cardiology, was interested in urokinase receptor. And what was interesting is for us, and why I am mentioning this example, because we generated an mRNA which coded for this urokinase receptor, and this protein, which is coded for this protein, the protein is highly modified, glycosylated, GPI linked, the carboxylator everywhere is modified. And what was interesting is we delivered the RNA inside the cell, and the cell knew what to do, how to decorate, and made it functional. So at that point, we thought that it could be useful for some kind of cell therapy. But again, I have to leave this position. I have to say that I wouldn't be here if I wouldn't be terminated in my position several times. So now that I moved to neurosurgery, still at Penn, and I worked 17 years in this laboratory. And we tried to develop messenger RNA treating a patient who had a stroke. And interestingly, again, I work with a physician who visiting the patient, and I am in the laboratory, and we try to educate each other, find a common ground, and then develop something. So we did a lot of improvement here, but we were, again, was not aware of certain things which I learned when I met my fellow awardees, and Drew Weissman, and you will hear from him more. So that Drew was coming to University of Pennsylvania, and he wanted to develop a vaccine. I was not interested in vaccine, but I was interested to help and to work together. And Drew generated these dendritic, human dendritic cells. And when I made the RNA and he exposed to it, you know, everything seems that the RNA is the perfect vaccine, as all of these markers were upregulated, and also found that inflammatory cytokines were increased. So it was not a good news for me. I was not very happy about inflammatory molecules. And we were thinking with Drew that what could be the reason, because the RNA, I assume that what I'm making is the same as inside the cell. So we thought that because this is immune cells, and maybe because it's coming the RNA from outside, it's recognizing as a danger. And to find out that whether if we isolate RNA out of our cells, whether it will be also immunogenic, similarly what I made in the tube. So for that, we did this experiment, and we isolated RNA from different compartments of the mammalian cells. Here is in purple what I synthesized in the tube, and you can see that what we measured on these dendritic cells is the inflammatory molecule called TNF-alpha. So I made the most inflammatory molecule, and what we isolated was not as inflammatory. Interestingly, the tRNA was not induce any TNF-alpha at all. Knowing that transfer RNA has a lot of nucleoside modification, this gave us the idea that maybe modification is needed to suppress immunogenicity of RNA. So we were thinking that okay, what kind of modifications are there? And there were already a lot known is present, more than 100 modifications in our RNA in the purine and pyrimidines. The only known modification, which enzyme, because we couldn't just order an enzyme to introduce and prove that really modification can suppress immunogenicity, the only modification was known 2' methylation and pseudouridylation, and this was discovered by Tamás Kis, is a fellow Hungarian student from the same university, same dormitory. We lived and studied and together, and so I called up Tamás and asked help and told him that, can you send those things so that I can change and modify the RNA? And because I was nice to him, so he responded, but he said that he cannot send because really these things require special structural element, and I don't have in my coded RNA those. So then I call up János Ludwig and ask him that where I can buy nucleoside-modified RNA, nucleoside-modified triphosphate, because the other idea was that maybe we have to purchase those and then try to see that whether we can incorporate. So we purchased 10 of those, which is present in the human body, and we were testing out whether we can make RNA. And 5 of them, these incorporated to the RNA. So now that we had nucleoside-modified RNA are ready to test that whether it is indeed is responsible for immunosuppression. What we found is that some of these were still immunogenic, some of these RNA, but others were not immunogenic, doesn't induce any TNF-alpha. When we made a closer look, we found that all of these actually modified uridine-containing. So we concluded with Drew that if uridine is modified in the mRNA, then it is not immunogenic. Of course, we wanted to make whether vaccine or I want to make therapeutic protein encoding RNA, so that we needed that it will code for a protein. So the next step was to see whether it can be translatable. And well, 2-thiouridine modified was not translatable at all, but when we use the pseudouridine-containing RNA, we get 10 times more protein on it. So it was now is a very good news that we get non-immunogenic RNA, which can be translated. I have to say that we describe also that Tol7 and Tol8 was actually activated uridine-containing RNA, and later, 10 years later, it was demonstrated crystal structures that actually the uridine, the nucleoside itself, can help to dimerize toll-like receptor 8 and activate it. And also when we did with experiments, we didn't know that our mRNA has pseudouridine, but turned out that again, 2014, several papers published that our RNA also has pseudouridine in it and incorporated by different mechanism. What we have done, we did after all of our discovery, what usually in the United States you do, you file a patent, then send in the paper, and then establish a company. We established RNARX, and we submitted also a small business grant, and we proposed that we will deliver to animals erythropoietin-coding RNA, and we will demonstrate is that it can increase the hematocrit of those animals. And that was a proposal for treating anemia with mRNA. So we generated mRNA, as coding here in the red, shown pseudouridine-containing RNA, and uridine-containing RNA, and we also injected just erythropoietin to the animals. And this protein half-life is very short, so it quickly disappeared. But what you can see here that pseudouridine-containing RNA, when we injected and translated to erythropoietin, we could measure the protein for 4 days. So it was extended translation of this molecule, which otherwise has 2 hours half-life. And it was physiologically relevant, and it was biologically active because it increased the hematocrit. And when weekly we injected, then we could maintain that high level of hematocrit in the animals. And it was important to demonstrate that pseudouridine-containing RNA even in the mice was not immunogenic, did not induce interferon. So this was the point, again, that I had to change, was forced to change my position, and then ended up in Germany at the BioNTech. At that point, we were not in this fancy building, we were on the campus, and BioNTech had no website. I just accidentally learned existence of it, but I went there and joined forces with Ugur Zahin and his wife, Özlem Türeci, and first we wrote a big paper about what you should know about mRNA, and we further improved the mRNA. You can improve the RNA by codon optimization, purification, nucleoside modification, many things. So as a result of all of our work we did with Drew, we did here in BioNTech, instead of this small amount of protein, short period of time, now we could make a lot of protein for extended time. So the mRNA was ready for prime time and test out. And my first project at BioNTech, we did messenger RNA. This contained 1-methylpseudouridine and coded for bispecific antibodies, recognizing one part is the immune cells, the other part is the tumor cells, and bring close proximity. We could demonstrate that such bispecific encoding mRNA injected into the mice can eliminate already established large tumors. These studies is presently in human trial. The other project we did when we injected mRNA to the mice, and this was again a cell surface closed tumor, so that like this is also in human study for head and neck cancer and melanoma, and these tumor were injected with mRNA coding for these cytokines, and these made the cold tumor hot, immune cells rushed there, and then when it circulated, they could eliminate the remotely located tumors, so that when it was injected these mRNA coding these cytokines, the lung had from the lung eliminated these tumors. So while we were working on this project in BioNTech in Germany, my colleague Norbert Pardy in Drew Weissman's lab generated vaccines using nucleoside-modified RNA. This was 2017, and that time the Zika virus was a major problem, and what Norbert was doing, generating mRNA coding for this envelope encoding protein of the Zika virus, and this was 1-methylpseudouridine, the nucleoside-modified RNA. The formulation was by Acuitas, the same as was for the BioNTech Pfizer vaccine, and of course we know that, and you will hear from my fellow awardee, Peter Kulis, about importance of the lipid nanoparticle, which contains different elements and were important adjuvant activity. And what happened is when these were injected to the monkeys, very small amount could protect the monkeys from Zika viral infection when it was challenged. So even 50 microgram it was a very revolutionary, because I have to say that prior to that, all of these DNA vaccines which were used unproportionally had to give higher dose to when the animal size was large. And so in human case, it was already milligrams had to be injected, but not in the case of mRNA. So this paper actually was selected for BIAL, the most important paper in the last 10 years. And so this was served the basis for developing the vaccine. And you are already quite familiar how we, 2020 January, we learned the sequence information. And by the end of that year, we also get the vaccine. So if all of these things would happen even just 20 years before, information would not have been enough to receive, because 20 years ago, you just couldn't order DNA synthesis, so the genes couldn't be ordered, but here you could order, and of course, with the help of input from government and others, it accelerated and helped to run the clinical trials, and that, of course, many people thought at that point that this was the first time nucleoside-modified mRNA was injected in human. What you might not be aware of that 2018 messenger RNA, nucleoside-modified RNA, was already used in a human trial for heart failure, patients who are undergoing bypass surgery. The RNA in 30 different point was injected to the human heart, and that RNA coded for VEGF-A, which is a protein that generates new blood vessels and then increase the blood flow in a part of the heart. And this was already in phase 2 trial and was very successfully completed, and the patient who received the mRNA felt much better. And other trial was, which was again using nucleoside-modified mRNA by Intellia. This was a Cas9 mRNA, and this was treated for patients who get amyloidosis, the disease. Here, the mRNA coded for Cas9 protein, as well as the guide RNA was delivered in the patient infused, and this toxic protein made in their liver cells. And so what happened is, with this technology, the genes where the toxic protein was produced was interrupted. And it was only one injection was sufficient that a patient, even 1.5 years later, were not producing any toxic protein. So this was very successful and ongoing. And right now, many, I don't talk about the vaccines, but for vaccine messenger RNA, nucleoside-modified RNA is used for now that not just for COVID-19, that's approved, but there are CMV, RSV, other viruses, HIV, and influenza, and so on. But I was always interested in the protein replacement therapy when the mRNA code for therapeutic protein. And those mostly, of course, secreted products were tested at the beginning, and this were the VEGF-A. Monoclonal antibody, BioNTech has one, not just a bispecific antibody, but one mRNA recognizing cancer-specific proteins, and cytokines, which was also injected in the, whether in the tumor or otherwise. And a lot of preclinical, I don't even have time updating all of these because so many trials, so many new companies are formed and proceeding with that. And I am very grateful for my colleagues who I work with at BioNTech, mostly because prior to that, I never get an R01, so I never cannot say thanks to the graduate student and the postdocs because I didn't have one. And so most of this experiment I did with my own hands. And I am also very grateful to my family. My daughter is a 2-time Olympic champion rower. So I am telling to the girls here that, you don't have to choose whether you have a career or a family. Here I am, an example. And thank you very much for your attention.